Okay, so we're going to move into D3 now. We're going to start with some of the standard level stuff. So it's pretty easily chopped in that the end part on linkage and dihybrids will be HL only, and the rest is going to be all SL as well. So the majority is SL, and then we'll tackle the HL part at the end. So I'm going to basically do a little bit of terminology and background first, and then kind of a big chart basically going through the different patterns of inheritance, how to write them, what they would look like on a pedigree. I'm not doing a ton of monohybrids everywhere, Punnett grids. So if you are still struggling with the actual problems, you'll need to go back and do some practice problems. This is really the theory behind it. So this is making the assumption that you know how to do Punnett squares, and we're just going over kind of the theory background. All right, so like I said, I'll do a little bit of history and terminology to start us off, just because we don't want to miss those little parts. So in terms of history, just need to know a little bit about Gregor Mendel in terms of his role. So again, we're just going to kind of clarify that the history was that it started with Gregor Mendel in the kind of 1850s era. And all of his work was done using artificial selection. So that's very clearly stated in the actual course outline. So we want to recognize that. So he used artificial pollination. And I think that term is probably fairly self-evident. So crossing pollen from one plant, putting it literally onto a stigma of another. So usually removing the anthers of the female plant and then placing the desired pollen there. And his work was done with peas. He was living in a monastery, so it was essentially what was available to him. Other kind of terminology stuff, which isn't overly challenging, but just making sure we're good on the generations. So when he did these crosses with peas, he basically started with purebred parents. So they were always lineage with one trait. Then he would cross the purebred parents, and that would give rise to a hybrid F1 generation. And from that hybrid F1 generation, he was able to essentially determine this concept of dominance. So the idea that the hybrid F1 tended to have the same phenotype, or they did in his crosses, as one of the purebred parents, kind of developed that concept of dominance. And then he would cross these with each other. And then you would end up with an F2 generation. And his most significant finding was that while the hybrid generation would be 100% one phenotype, the F2 generation would have the 75%, 25% ratio. So this idea of essentially the resurgence of that 25%, so re-seeing this kind of lost phenotype, led to this concept of dominant and recessive alleles, and that the other allele must still be there. It's just not being seen in the population at the time. Okay, so that's just a little bit on that background stuff on terms of Mendel himself. Then let's do a little bit of terminology. One thing a lot of students are quite good at doing the Punnett squares. IB uses Punnett grids. I'm going to try to remember to sometimes use that term, but it seems strange to me. But most students get quite good at doing the crosses, but then again, it's the terminology that gets a bit tricky. So we're just going to make sure we're really good on that. So when we see single letters like that, we are looking at alleles. And when we say alleles, we are talking about versions of a gene that are different because of single nucleotide polymorphisms. So they're generally almost the same except for single nucleotide differences. So we're just going to write different versions due to SNPs, which is single nucleotide polymorphisms. I just don't have the room for all of that. So single nucleotide polymorphisms, single base differences. And so again, we said that he was able to use this findings. So essentially from the F1, that was what was used to determine what was the dominant allele. So the dominant allele is going to be the allele that is seen in the hybrids or the heterozygotes. And then what shows up here in the 25% is the still present recessive allele. So that's those terminologies. So from alleles, we end up with genotypes. So alleles is a single letter, and this is all we did this in kind of Hardy-Weinberg as well. So being clear on alleles versus genotypes. Then we have genotypes, which is like big A, little a, big A, big A, big A, little a, what I wrote. So these are going to be our genotypes. And when we say genotypes, we just mean the allele combinations. So what combination of alleles are there? Terminology, again, here is going to be important. So we're going to say that both of these are homozygotes. You can also see the term purebred being used. Often purebred is used for the phenotype, so it's just saying coming from a long line of the same phenotype, whereas homozygous is if we can confirm it by genotype. And then we have the heterozygote. We want to also recognize terms like hybrid or carrier and hybrid. So those are all different things that would mean that. Okay, then the key here is that the next thing we need to look at is going to be phenotype. So then we would have like tall or short. So alleles, genotypes, phenotypes. So alleles cause your genotype. Genotypes lead to the phenotype. And so when we say phenotypes, these are going to be the traits. So these are going to be in some way an observable trait. It's not always externally observable. So it could be like your blood type. So I can't look at someone and be like type A, but I wouldn't have to do genetic screening. I could do some sort of blood type testing. So there would be ways of determining the trait that way. So that's what we mean by phenotype. We need to add something here though. When we say phenotype, phenotype is going to be genotype plus environment. So it's the combination of the alleles you have and kind of environmental influences. Also want to know, how does the environment have an impact? Often by affecting gene expression. So how the gene is used. So we know what the gene is, but how often, how regularly, kind of patterns of its expression or use. And then just a terminology thing in our little terminology section is the term phenotypic plasticity. And that actually refers to essentially the ability to change expression based on the environment, and it can be adaptive. So it can be an adaptive tool that basically says these genes are expressed in this environment because in some way that benefits the organism. So phenotypic plasticity is that ability for phenotype to change in some way based on environmental conditions. Good. One more thing I wanted to emphasize here is just looking, and we'll get to this a little bit more with HL in terms of the laws, but recognizing that with alleles there is only one passed on per gamete. So that in HL terminology is essentially going to be the law of segregation, that only one copy of each gets passed on, so the two alleles will split. And then, of course, there are two copies in diploid cells. So there's always two alleles in the diploid cells, which is what gives you a genotype. So one in gametes, two in the actual cells post-fertilization. Okay, so like I said, that was just my little bit of making sure that we know the terminology. So what we're gonna do now, and this is gonna almost take us through our SL part, is we're gonna do a pretty big chart looking at the different patterns. So I'm gonna erase just so I have plenty of room for that. Okay, so what we are looking at now are the different patterns of inheritance. So that's kind of our background information. So there are different kind of patterns that we see. In class, I distinguish these as Mendelian, non-Mendelian. That terminology isn't really essentially in your course, so in your syllabus. Basically, everything other than dominant, like complete dominance, is non-Mendelian because all of his patterns fell there. So we're going to look at patterns of inheritance, and then we can, once we've determined the pattern, we are then going to solve the problems with Punnett grids. So again, I'm not drawing a bunch of these. I'm only gonna really do the dihybrid ones. Dihybrid ones are a bit trickier. I'm not gonna actually draw monohybrid ones for each trait. They're pretty straightforward in terms of working them. So I'm just drawing one as a little reminder of a monohybrid Punnett square, Punnett grid. So again, we're just looking at like big A, little a, big A, little a. Okay, so that's kind of the tool that we're going to use for all of these next ones. They could all essentially be thrown into a Punnett grid, a monohybrid one. Okay, so for our chart, we're going to say pattern. And this can go all the way across. It did for me. Then I'm going to say description. Then I'm going to actually put the letters. This is important for some of these, is making sure you use the right notation. Then we're going to say, is there an example written into the course? So on a lot of these, the syllabus says, for example, and names it. So we're going to put that there. And then the last thing we're going to put is there a pedigree pattern that we should know? There's a couple instances where that's not relevant, so when relevant. So we're gonna look at kind of the pattern. So by doing this, I'm kind of merging pedigrees into this, so rather than doing kind of a whole separate thing on it. Okay, so for your information, we're gonna have 6 total rows. So we'll work on to that as we go. So the first two I probably could have put as 1, which is essentially just complete dominance. The difference is I'm going to break up autosomal dominant, autosomal recessive. Those are both the same pattern, which is complete dominance. The difference is whether the disorder is one or the other. The difference really only matters for the pedigrees, but I'm going to essentially clarify that here. So I'm going to put them separately. So I'm going to say the first pattern is autosomal recessive. So when we are looking at autosomal recessive, this is also the same kind of thing as saying complete dominance. So when we talk complete dominance, what we are looking at here is that the heterozygotes have the same phenotype. So the heterozygotes have the dominant phenotype. So that is going to be true anytime we are talking about complete dominance. What makes it an autosomal recessive trait is that you need 2 copies to have the trait. So in this case, you would require 2 copies to have the trait, disorder, whatever it is. Okay, so it requires the 2 copies. We're going to move this over a little bit because this can be a small one. Okay, so in both of these cases, we use just the standard notation of big, little. So we're going to use big A, little a. And again, if we are talking about an autosomal recessive disorder, little a is what represents the disorder. Okay, examples. There is a named example they want you to know for this. So the example is PKU. So phenylketonuria related, but you can just use the abbreviation. It is due to a faulty enzyme. And the faulty enzyme was supposed to be turning phenylalanine into tyrosine. And the syllabus does actually specify that. So possibly a little bit of extra info you need to know. So essentially that enzyme doesn't make that conversion. So high phenylalanine levels end up causing it. Okay, what are we looking at on a pedigree? So on a pedigree, we are looking at the pattern whereby we see carrier parents go on to have a child that has the condition. So we're seeing unaffected parents have an affected child. If that affected child is a daughter, we can confirm it's autosomal recessive. If it was a son, we would also want to check gender ratios. So equal male to female affected. So we don't see any difference in the number of males and females overall, no significant difference. So you might have slightly more of one than the other, but there's not an overwhelming gender pattern. Okay, so that is autosomal recessive complete dominance. Next one, like I said, is essentially the same pattern, but if we're looking at it for a trait or disorder, this time we're going to call it autosomal dominant. And this is also complete dominance. Comdom, that's what I mean. So then that also means that this is the same. So the heterozygotes also have the dominant phenotype. So same idea here. But in this case, you only need a single copy. So essentially one copy is required for this trait or disorder, which means heterozygotes have it. So the only difference in these two categories is whether the trait we're tracking is the result of the recessive allele or the dominant allele. So again, we're going to use big A, little a, but this time the trait we're tracking is marked by the big A. There is not a named example that you need to know for this. So you can just put a dash. If you're interested, Huntington's disease. I don't think it comes up anywhere else in this course, but that's probably the most commonly stated one. So it's just good to know in case it does come up. The other one is achondroplasia, which is dwarfism. A specific type of dwarfism is also a dominant condition. So those are some well-known ones, but none that are named in the course. And then this time we're going to see it in every generation. So there's going to be no skipping of generations. And in particular, we would be sure we are looking at dominance if we actually saw the reverse of above. So if we actually saw two affected parents able to go on and have an unaffected child. So if that was the case, that would be confirming for us that it is definitely dominant. Otherwise, if it's in every generation and everyone who has it has a parent who had it, you could probably presume dominance. But this would be for sure. And again, males and females would need to be equal if it's going to be autosomal. Okay, next couple of ones are going to be looking at kind of adding another phenotype for the heterozygote. So these actually are usually not shown on pedigrees because generally we use pedigrees when there's two phenotypes, so shaded, not shaded. We don't generally use them for multiple phenotypes, or if we did, we'd have to be using different colors. So we're not going to worry about that. So this next pattern is going to be incomplete dominance. So for incomplete dominance and same for codominance, which we'll do next, the heterozygote gets its own trait. So the heterozygote actually gets an extra trait. So the heterozygote has its own phenotype. And in this case, the phenotype is like a blend or an in-between. I'm just going to make a little note. I'm actually probably going to put them on the same one because codominance, I'll leave it for the next. Sorry, codominance kind of works with multiple alleles, so we'll put them together. Okay, so the heterozygote has its own or in-between phenotype. So for this, we're using subscripts. So we're using like color red, color white. So we want to show that neither of them is dominant or recessive. But we want to show them on the same letter so that we show they are one gene. So that's the way we're going to notate that. You do have one that they want you to know, which is the red, pink, and white flowers of the marvel of Peru. Or sometimes it's called the 4 o'clock flower. I doubt you need to memorize the name. And I think if it stands out as pink, you should be able to remember incomplete dominance. So we actually generally do not use pedigrees for more than 2 phenotypes. So when we have greater than 2 phenotypes, we generally do not use pedigrees. Again, you can, but you wouldn't be getting them on a test because the pedigrees we've taught you are to distinguish between 2 phenotypes. Okay, the next one I'm going to put together, although in theory they could be separated, but we're going to look at multiple alleles and codominance. So the example you need to know is an example of both, so that's why we're putting them together. So when we say multiple alleles, it means exactly what it sounds like, is that there are more than 2 unique, I'm trying to think of a good way of saying this. So the tricky part, I'm just going to explain this verbally. So we can have 2 alleles, and one of the alleles can have different SNPs that cause it. So different single nucleotide polymorphisms can cause the same allele as long as the protein outcome is the same. So as long as the defective protein is the same for all the different SNPs, we would still call it one allele. So if it's multiple alleles, it means it's multiple different protein impacts. So that's just a little bit of a thing to clarify, is that same allele doesn't actually mean it has to be the same allelic, like DNA-based difference, but it means the protein impact is the same. So more than 2 unique alleles is what I'm trying to say there, and that's what multiple alleles means. And then codominance, we're saying that the heterozygote shows both phenotypes, so it fully expresses both. So if this were something like color, it would be like red patches, white patches. So we see both full expressions in different places. The example we are going to use is blood type. It comes up a lot in the course, so I would be prepared for a question on it because they bring it up both when they're talking about glycoproteins and then again in the immune system and again here. So anything that shows up a lot is a good sign you want to know about it. So what we're adding here is the alleles. So we do want to use the I's, so IA, IB, and little I. So IA and IB are our co-dominant blood types, so they are both dominant. So again, our example here is ABO blood types. The other thing that it wanted you to note is that ABO blood types are an example of a discrete trait. And so what we mean by a discrete trait is that you can't have like somewhere like a little bit of A. You don't have like major A. You fit into a distinct category. So this discrete trait or another way of looking at it is categorical. So we could basically take people and evenly divide them into groups. So we can say this many have this many have this many have that. So it's an example of a categorical kind of trait. Okay, and again, we would not use a pedigree to solve these because it has too many different blood types, so it wouldn't really work for that. So that is an example of something we would not use. I did my box. I'm just gonna, this is not particularly relevant, but you can show discrete info with a box and whisker plot. So we could be looking at like a box and whisker plot of type A people, and we could say for people, individuals with type A blood. We could be looking at how many there are. We could be looking at how many infections they get or something like that. So we could be taking a group and then representing data within that group. I'm gonna put this here just because I don't know where else to put it. And I don't have to do a whole thing on it. So we're just gonna draw a little mini box and whisker plot here. Like so. And we're just gonna label a few key things on it. Like I said, this is just where the course has chosen to put it. So here we have the median. We have, if this is starting at the low, this would be Q1, this would be Q3, this would be the minimum, and this would be the maximum within the rule. So the minimum is always going to be, and then this would be the IQR. So again, the minimum is going to be Q1 minus 1.5 IQR is how you get that minimum. So it's not necessarily, or the lowest number, but it's not necessarily the lowest number. That would be the cutoff for the minimum. And then anything that is outside the minimum or the max is an outlier. And again, you would use this 1.5 IQR to work that out. Okay, so that doesn't really have anything to do with blood types, but I just didn't want to leave a little bit of space not used. So that's why we stole that space for that purpose. Okay, so that was just to go over something that has been allocated to this topic. The next one, which is another important one, and then we'll finish with polygenic or continuous, which is a pretty dot point thing. But the next one that is important is sex-linked. So if we are talking about a sex-linked trait, this is now going to be a trait that is on one of the biological sex-determining chromosomes. So it is a gene that is located on X or Y. Usually X because it's much bigger. So that is why it is usually X, and because everybody has at least one. These are going to be inherited differently due to the fact that XX is females, so they get 2 copies. And XY is biologically male, and so they only get 1 copy. So they are not, they're actually called hemizygous, but that word doesn't appear to be in your syllabus. But they can't be homozygous or heterozygous because they only get a single copy. If you are writing these, you have to write the X in. If you don't write the X in, your Punnett square will not give you the right result because then you'll get confused on the fact that they don't have another copy. So you have to write these with the X. So X big H, X little h. The example that you are expected to know in the course is hemophilia. Hemophilia is an X-linked recessive, and it is linked to clotting factors. So again, we did clotting factors in C3. So we know that that's part of the pathway that ultimately builds fibrin. So it's a defective thing in that. Okay, so what do we look for in the pedigree? So rather than drawing them, we're just going to write kind of the 3 possible rules. So in the pedigree, what we would be looking for is if we saw all males affected and they also match dad, then we would say that is a Y-linked trait. Again, there's not very many of them, so I feel like if they included that, it would just be as a bit of a gotcha to make sure you don't accidentally say sex-linked recessive. If we just see more males affected and they're not necessarily all matching dad, then that would be X-linked recessive. Notice I didn't say recessive or dominant Y because there's only one copy of it. If we actually saw significantly more females affected, then we would actually look at the possibility that it is X-dominant. These are just good things to look at before you make a final conclusion from a pedigree. So the other kind of wording they put in there is the whole inductive deductive. So inductive is when we look at data and try to come up with a hypothesis. So that's kind of what we're doing when we're looking at a family dynamic and then saying, I think it's X-linked. So we just want to make sure that we're checking all of it and not making an induction based on one little family, but we're trying to look at all the information because we're then going to use our presumed pattern to deduce what we think that means about people's genotypes. So we want to try to give a really good look and make sure we don't miss anything. Okay, last line that's on here is the idea of a polygenic trait. So a polygenic trait. The other term you'll see there is continuous variation. We'll put that in a minute. So in this case, when we're looking at a polygenic, it means many genes contribute to one trait. So it's usually like an additive effect. So when we go into dihybrids, we're just looking at two different traits, so two genes, two traits. Here we're looking at more than one gene adding together for a single trait. So our key distinguisher here is that they're all working together on this single trait. You don't really need to worry necessarily about notation because you probably won't get a problem with these. But if you were to do this, you would just use different letters for all of the genes. So you don't have to use, there are different genes, so it would be sensible to use multiple kind of letters. They do want you to know that skin color in humans is an example. And this is a continuous trait. So it is not categorical. You can't just evenly divide people. So height's another good example. So there's people kind of all along the scale. So you can't group people into distinct categories. And one tool that you might use to show this, rather than doing like a box and whisker plot, which tends to be for a distinct group, if we were looking at this, we would have to look at more of like a normal curve. So what you might see here is this might be shown with a normal curve. Again, we would generally not do a pedigree because there's too many different phenotypes. Okay, so those are essentially the 6 patterns that you need to know what they are. You need to know what letters to use. You need to know what example the course gave you, and then you need to know how to make sense of them on either a pedigree or just on some sort of data set. So it might be a box and whisker or a normal curve instead of pedigree, but how to actually show them. Okay, so I had one tiny little point, but I've totally run out of room. So SL, stay with me for 1 second because I want to write one more thing. And then we'll move on to the HL stuff. All right. So I just wanted to quickly look at these, just write one line going through these 3 different tools, just so you have some idea of when you would use them. So this is my last line for SL, and HL will just put it under the chart. look at different tools, I want to look at pedigrees, Punnett squares, and box and whiskers. Those are your three kind of problem solving skills. So when we look at pedigrees, what we are doing there is we are inducing. So we're going to induce a pattern and then use that to deduce genotypes. So that is the purpose of a pedigree. So the reason why we would use a pedigree is to be able to say, here's a pattern, what do we think is going on, and how does that affect different people? Then if we want to predict offspring ratios, so if we want to predict, so this is induce and deduce, if we then want to predict genotype and phenotype ratios, I'm just doing abbreviations, then we're going to use a Punnett grid. And some problems may make you do both. They may start with a pedigree. From the pedigree you do this, then you set up a Punnett grid. So then we're going to use a Punnett grid. And then the last one, again, that doesn't really fit super well, but the last one is if we wanted to actually look at trends. So if we want to show what kind of trends are going on with different traits, then we might use a box and whisker plot. So if we wanted to show trends by trait, so what's the trend for this group versus this group, then we might use a box and whisker plot. So those are kind of your three tools that showed up in this unit that you need to be able to kind of utilize. Okay, that's all SL, so bye-bye. HL, let's do some dihybrids. So we are gonna now look at dihybrids, and we're gonna basically look at unlinked genes versus linked genes, and at the bottom just remind ourselves of how do we know which it is. Okay, so I'm just gonna erase all of it. All of it. Okay, so now we are moving on to dihybrid crosses. Okay, we're going to make our little reminder that this is HL only. And so what is distinct here, it's different from like a polygenic trait, is that we are now looking at two genes and two traits. So they're two separate genes, but they're also two separate traits. So we are considering two genes that cause two distinct traits. They may be somewhat related, and maybe hair color and hair curliness, so they might be related to a similar part of the body, but they are distinct traits from one another. And even like the example of like production of pigment, and then the other one would be kind of color of pigment. Those are still technically separate traits. Are you able to produce pigment? Okay, what color is it? So two different traits are two different proteins. So they're making totally different proteins. Okay, so we are going to then divide our space into two. And then we're going to leave some room at the bottom. So we're going to write small, depending on how much room you have left. We only need a tiny amount at the bottom, so you can use the rest of your space how you have it. Okay, so I want to look at unlinked versus linked genes. Okay, so if we have two different genes, basically we're looking at, do they act independently of one another, or do they in some way stick together? So we're going to look at the idea that these are genes that independently assort. So these are genes that independently assort. And then the alleles separate independently of each other. So which allele you get for this one has nothing to do with this one. So alleles separate independently. So while we're on this point, I just want to add a couple things in. First of all, these are laws that Mendel himself saw. So he did actually recognize this. So he said the law of independent assortment, which is that one gene does not impact another. So the idea that the genes are unrelated, so to one another. And then also said the law of segregation, which was just the idea that alleles have to separate. So alleles have to separate, and then independent assortment means separate to one another. So they do have the laws in there, so that's like a very short little reminder of that. The other thing is that Mendel didn't know this, but we do know the actual meiotic reason why. So we understand that independent assortment is because chromosomes lining up separately in metaphase. So we know that that is going to happen if they are on different chromosomes. So if they are on different chromosomes, we know that the chromosomes are going to line up independently at metaphase, and that's what's going to cause this. The actual segregation then is in anaphase. So they line up separately first in metaphase, then separate in anaphase. Okay, so again, our first assumption is that they are on two different chromosomes. We also understand that if they are on the same chromosome, but really far apart, they're also going to act this way. So the first thing that makes them unlinked is different chromosomes, and then we'll add in a minute, or actually go ahead and add it now, or they can be on the same chromosome, really far apart. So if they're on different chromosomes, the reason they assort independently is because of this lineup in metaphase. So the reasons do differ. So in this case, it's because of the chromosomes lining up differently in metaphase. If they are far apart on the same chromosome, the reason they independently assort is because they are separated by crossing over all the time. So if they're really far apart, crossing over just separates them so often that they essentially act the same as if they're on separate chromosomes. So both of these are actually the same. Sorry, probably confusing writing it here. But either of these can cause unlinked genes. So different chromosomes or far apart. Okay, so let's see how we can actually solve these. So when we are looking at this, so genes that are independently assorting, we are going to solve these problems with 16 box Punnett squares. So a reminder that you can do shortcuts. So I'm going to go do a full one and a shortcut one, just to remind you of that, but know how to do a full one in case it asks for you to work it out in like a longer response question. Again, new course, we're just not exactly sure how these things could look. Okay, so let's say our first example is the bad one. Big A, little a, big B, little b, big A, little b, big B, little b. So reminder that you can use FOIL for gamete combinations. Okay, so I'm making these pretty small guys. I'm sorry if I drive you mad with it. I'm not going to actually fill it in. I'm just going to set it up. So that's why I'm making it pretty small. Okay, so there's our 16 box Punnett square. When we say foil, we mean first of each letter. So big A, big B. O means the outside ones. So that's big A, little b. O, I is the inside ones, little a, big B. L is the last of each. Just to show you that again on the other parent. First A, first B, A, big B. Outside, big A, little b. Inside, little a, big B. Last of each, little a, little b. Okay, like that. You can memorize this, so you can actually now solve this, but I strongly advise you memorize this phenotype ratio. And it's going to be 9 dom, so dominant for both, 3 dominant for one trait, recessive for the other, 3 swap, recessive for the first one, dominant for the second, and 1 recessive for both. You do need to know what those actually mean. Okay, so that is one that you would need to know. The other one that I would recommend you know, I would recommend, the syllabus told you you should know, is this one here. This is what we would call a test cross, and they're often used to determine linkage. So we'll refer back to them in just a second. But where we use a double homozygous recessive individual. And in this case, we shouldn't even totally need to FOIL, but you can actually shorten this and only do a single row. So this is just to show you how you can take some shortcuts. So for the first one, we would FOIL again. First A, first B. Outside letters, big A, little b. Inside letters, little a, big B. Last of each, little a, little b. We don't really need to FOIL the other one. If you FOIL it, you can. You're just going to get that they're all little a, little b. Okay, so and then in this case, each of those will have a different phenotype. So the phenotype ratio here is 1 to 1 to 1. As we'll discuss in a minute in terms of using chi-square, if you know these ratios, you can save yourself a significant step in a chi-square problem, so definitely worth knowing. Okay, so that is unlinked. Unlinked, one has nothing to do with the other. Whether you give a big A has nothing to do with whether you give a big B, hence why there's 4 different gamete combinations. Okay, so then what do we mean by linked? So now, if they are linked, these are close together on the same chromosome. Trying to see how my spacing's going. So that's important because the same chromosome is crucial. So the same chromosome means that they are in the same place in metaphase. So they stay together in metaphase. So they're not going to be shuffled because they're on the same chromosome. So they're going to stay together. The reason why the close together is important is because that means that they only get separated if the chiasma happens to fall between them. So they are going to usually not be separated by crossing over. And the only instance where they would be is if the chiasma falls between them. And if they are close together, that doesn't happen very often. So it can happen, but not very often. So the closer they are together, the even more likely that is to happen. Okay, so we do need to be able to identify when it has happened though. we want to determine if that occurred by using recombinance. Okay, so if we are doing this with recombinance, first of all, we want to look at how we would write this. So we would look at it, and this time, we would write it like this. So instead of writing it like that, we're writing it this way. So that's actually showing these were on one chromatid, or chromosome, this was on the other homologous chromosome. Then we would do a test cross. The reason we're doing a test cross is because what we want to do is we want to focus on this parent. So we want to actually look at this one. So this is what we're focusing on. But we're using this one because it's going to be masked. So this way we can actually look and tell from genotype or phenotype what's going on here. Okay, so then let's look at what that would actually look like. So if we were to actually look at the gametes, what we would see is if they stay how they are, you would have big A, big B. They stayed on the chromosome together. If they shuffle and you see big A, little b, that tells you something crossing over got in the middle of it. If you see little a, big B, that tells you crossing over had an impact. Little a, little b, that's in an original combination. So what you would see is that these are recombinants due to crossing over. And we would expect, if it is a linked gene, that we would expect these recombinants to be rare. So we would expect the numbers of these to be much lower than if you were seeing these originals. So these, on the other hand, would be parental combinations, and you should see lots of these. So you should see lots of the two parental combinations and very few recombinants. So you might see some because of crossing over, but not very many. And we can do this by alleles, or you can also determine by genotype or phenotype. So if we're looking at genotype, we remember that this one has no impact. So we would be looking at like big A, little b, or the other parental, which is little a, big B, little b. So again, that just means you need to include the little a, little b from the other parent. Or we could do it by phenotype, depending what that looks like. When we're doing it at phenotype, we're looking at for new combos. So we're looking for new trait combos that we didn't see in either parent. So neither parent would have that combo. Okay, so that is essentially the theory behind what linked genes are. So they stick together more often. We don't see, so this would be a test cross. We don't see a 1:1:1 ratio because we see lots of the parentals and very few recombinants. So we may still see 4 phenotypes if we have a large number of offspring, but it's very biased towards the parental phenotypes. So that's what we would see. So then now we need to say, well, how do I know which it is? So how do we determine if it is linked or unlinked? So if we were to have 2 genes and we wanted to say, is it linked or unlinked? We would basically look at how the offspring turn out, and we would say, does it look like this? Are they fitting the ratios we expected? Or does it look like this, where we're seeing a lot of a couple and not very many of these recombinants? So we see that, and then we have to say, like, well, is it different enough? So we do that with a chi-square goodness of fit test. So goodness of fit rather than independence, but you don't really need to know that distinction. Okay, so when we do the chi-square test, the null hypothesis is that they do assort independently. So the null hypothesis is that it looks like this, or that there is no difference, no statistically significant difference between observed and what we would expect for independent assortment. So the null is always that they independently assort. That is the more likely situation because there are a lot of chromosomes and two genes are just statistically more likely to be on different chromosomes than they are the same. So, and particularly because we've said here for them to be linked, they have to actually be really close on the chromosome. Just a quick note, we'll go back into this, but at the end of a chi-square test, you're not going to be able to tell which of these it is. So you're not going to be able to tell if they're different chromosomes or far apart on the same. You can just say unlinked or linked. So we can't definitively say that they're definitely on different chromosomes, but we can say they independently assort. Okay, so no statistically significant difference between observed and expected for independent assortment. The alternative hypothesis is going to be that the genes are linked. So essentially the difference between observed and expected is too much to be explained by chance. Therefore, we could make the conclusion that the genes are in fact linked. Okay, so then what we need to do is we need to actually determine what is the expected. And since the expected is presumed on independent assortment, we're going to use the phenotype ratio from a dihybrid. So you're going to do the dihybrid, or you're going to not do it because you've already memorized your ratios. So use the phenotype ratio from the dihybrid and multiply each ratio by the number of offspring. Again, most of the time this is done with test crosses, so that's the more common situation. So what we would basically be saying is take the number of offspring and each phenotype should be 25%. So you should be able to go 25%, 25%. That's generally how it's done. They might make you do it with a dihybrid or a double heterozygote, just be mean, but generally you do it with a test cross. So you're going to take your phenotype ratio from the dihybrid, multiply it by the number of offspring, and that will give you your expected. Then you're going to use your expected and you're going to calculate chi-squared. Just a reminder that chi-squared is the sum of observed minus expected squared over expected. So that is our chi-square value, how we would determine that. Then we're going to use our DF is always going to be 3. I'm just going to write number of phenotypes minus 1. You can actually use these for like testing if something is incomplete dominance, but you don't have to do that in your course. It's only for this exact use. So your DF is number of phenotypes minus 3, which is essentially always should be 4 possible phenotypes. So essentially always use DF of 3. And then we're going to use P of 0.05 because that's the general confidence level that we are going to use. And then you are going to make your conclusion. So if chi, and we're going to do that to find the critical value. So then we're going to say if chi-square is greater than the critical value, then that means we reject the null and they are linked. If chi-square is less than the critical value, then we are going to accept the null and we are going to say therefore they are unlinked, or we can say they independently assort. So either way, so you could say it either way, linked or unlinked. Okay, that's everything on the genetic stuff. Good luck.